Hello, everyone. It's Gina. Um, it is Sunday. It is Passover. It's Easter Sunday also. Hope you celebrate are having a lovely day. If you're watching this on Monday, I hope you had a lovely day. I am going to make a few recordings for the genetics chapter to help you along. I'm going to make a recording of some of the vocabulary and a recording of how to do some of the Punnett squares so that you can have these to practice with. You're also going to go over the Punnett squares in lab, but this is a recording, so you can fast forward and play it and fast forward and play it and rewind it and all that good stuff. So hopefully this will be helpful. I actually just purchased an iPad and an iPad pen just so I can do this. So hopefully these tools will become helpful for you in the next couple of chapters, because the chapters coming forward have processes, unlike the previous chapters where these have real true processes that you have to kind of follow through. So hopefully this system, I'll make more videos, and hopefully this will help you. I'm in my house. I am not in the woods. This is Pittsfield State Forest. I took this picture last May or maybe early June. So this is not some special place anywhere in the world. It is literally 1 mile from the campus of Berkshire Community College. So you can go for a hike in the back there, and you would end up where this picture was taken, and it is absolutely beautiful. All right, so I'm going to share my screen and show you the PowerPoint for this chapter. So we're going to cover genetics, and really truly what genetics is the study of inheritance, right? It's really truly just a study of how do genes, right? How do genes get into offspring, right? So, well, they're in the sperm and the egg, right? The sperm has half of the copies of all the DNA for the organism, and the egg has the other half of the chromosomes for that organism. Those chromosomes are just big long stretches of DNA, right? That's all chromosomes are, big long stretches of DNA. And each one of those chromosomes contains genes. So for example, you have 23 pairs of chromosomes, right? And each one of those chromosomes has a bunch of genes on them. So for example, chromosome number 1 has like 2,000 genes on it. Chromosome number 2 has a little less, and then on until you get down to that 21st and 22nd pair. And they are really small, so they have less genes on them because, well, they're shorter, so they don't have as much DNA, so they can't have as many genes. So we want to remember that the information that's being passed from the sperm and egg to the offspring is going to be what that offspring inherits genetically, right? We know that there's more to this story. Really, truly, the environment plays a large role. Epigenetics plays a large role, but we're really not going to talk about that in this video. So what we're studying in this chapter is inheritance or genetics, right? Which is the study of how things are inherited. So I'm just going to go past some of these slides, maybe, if my computer will let me. All my friends who make flashcards, these are good flashcards for this chapter. Just briefly, Gregor Mendel is the father of genetics, right? We always say that. Gregor Mendel lived in the 1800s. He lived actually at the same time as Charles Darwin. But Charles Darwin and Gregor Mendel never met. If they had met, we probably would have a lot more biological advancements than we do today, because well, neither one of them knew what DNA was. You know more about DNA than Gregor Mendel or Charles Darwin knew in their lifetime. But what's interesting about Gregor Mendel is his research was not found until about like 50 to 75 years after he had passed. Somebody found a book on a shelf and was like, "What is this research?" I don't know if you can hear the puppy crying. And so Gregor Mendel, basically what he did was he studied pea plants. He was a monk. He lived in a monastery. He was a teacher also, and he studied pea plants. And what he learned from the pea plants is that every time he crosses them, there's like this strange predictable pattern that we see when he grew pea plants just like maybe you do in your garden, right? And he grew them up, and he would do it thousands and thousands and thousands of times. He had all of these pea gardens in Vienna where he lived, and every time he did these specific crosses, right, he crossed the plants, he always got a predictable pattern, and he wrote about that predictable pattern. And well, he didn't know they were called, he didn't know it was being crossed. He didn't know it was being transmitted from the pollen in the egg to the offspring, right? He didn't know what it was, but we know that they're genes now. We know that it's DNA, and we call them each individual packet of information a gene. And he called them units. He knew some kind of unit was being passed in the sperm and egg, sperm. When I say sperm, I mean pollen in this case, to the offspring. And he gave us this idea of dominance and recessiveness, that there's sometimes in a organism, in these pea plants, for example, there's a dominant trait that always expresses itself, and a recessive trait that is hidden unless the dominant trait is gone, right? And when I say that, when the dominant trait is gone, I mean like the DNA that is used to express the protein that makes that dominant trait is not there. The person didn't inherit it, right? They only inherited the recessive information. So he gave us this idea of dominance and recessiveness. And we were lucky that he used pea plants because they're really easy to study. They're called a model organism. And I'm just going to go forward and just show you a little bit about what he did, which was kind of cool. So this is the parts of the plant. You can read all about it. There's the male and the female parts are on the flower or within the flower. And you can see here that this bulbous thing inside here, this sort of looks like I don't know what it looks like a big wiffle ball bat. It's called the stamen. I mean, it's called the carpel, and the carpel is the female part of the plant, and that is where the ovaries are, right? All the eggs are stored down here in this sort of bulbous. And then the sperm producing part is here. This long stringy thing is called a filament. And this little end here, this little triangular yellow end here is called the stamen. And the stamen is where the pollen is. And the pollen will then, well, it can do one of two things, which is kind of interesting. In some plants, this is possible, but it's not true for all plants. In this case, what can happen is the pollen from this specific plant, this specific flower, can self-pollinate. The sperm or the pollen will actually has like a little enzyme at the end, like at the tip of its nose, that little pollen grain, and it creates this enzyme creates a tube. It eats the entire, like this little entry here to the carpel, and it'll come all the way down and fertilize one of the eggs inside. That's called self-pollination, and you're really creating a clone that way because you're using the same DNA connection each time. So you really, if this self-pollinates, you're only going to make purple flowers. So that's not what Gregor Mendel did. Gregor Mendel made it a little harder for himself, and what he did was he snipped off all the filaments. He chopped off all the filaments so the plants could not self-pollinate. And what he did was he crossed a purple flowered plant, pea plant, they're all the same species, and he crossed it with a white flowered plant. He chopped off all the filaments on this one. He made it so that they couldn't self-pollinate. And he used a paintbrush, and he meticulously took all the pollen from the white flowered plant and painted it onto the carpel of the purple flowered plant, so they cross-pollinated, right? So there was this connection of the DNA from the white flowered plant connecting with the DNA of the purple flower plant. So he was basically taking two purebreds, or what we call true breeding plants, and he was crossing them. Sort of, yeah. So I don't want to confuse anymore. So he just cross-pollinated them, right? A purple plant times a white plant. He cross-pollinated them, and then he let them grow, right? He said, all right, let's see what happens. Let's see what happens, just like in your garden, right? Let's see what happens when pollination occurs. And what happens is, and I always love that a lot of students find this amazing, and they don't realize this is how fruit forms. But what happens when fruit forms is the carpel swells, and it matures, and then it ultimately turns into like some kind of thing, whether it's like a fruit, like an apple or a tomato, or in this case it's growing into this pod, the seed pod, right? In an apple, the seeds are held inside that, within the fleshy part of the apple, right? In this case, what's happening is this pod is protecting the seeds inside, and we eat those seeds, right? We eat those peas right out of the garden. It's like the greatest thing. It's so delicious. But what Gregor Mendel did is he didn't eat them. What he did was Gregor Mendel took those seeds out because they are the production of the cross-pollination of a white flowered plant and a purple flowered plant. So he took the seeds out and he planted them. He's like, let's see what happens when we take the seeds from this cross-pollination and we plant them. What color plants will we get? We wanted to know. Are they going to be white? Are they going to be purple? Are they going to be striped? Are they going to be pink? Who knows? What are they going to be? And he did this thousands of times. And every single time he did this, he got all purple flowered plants. That is the basis of Gregor Mendel's genetics experiments. He went on and he did more, and he even looked at other organisms. But this is really the true basics of Mendelian genetics, right? Where he said, I'm going to cross these two plants. They have opposite traits. It's still the same gene controlling it, the gene for flower color, but there are two versions of the gene. One version gives you purple flowers, one version gives you white flowers, right? Same thing is true for eye color, although it's a little bit more complex. There's like 16 genes associated for eye color. But one version of all those genes together, let's say, gives you brownish colored eyes, and the other version gives you blue eyes, right? So it's the same gene controlling it. It's just which version of the gene are you getting, right? And in this case, what was happening is, as you can probably imagine, because maybe you had biology in the past or you've heard this word before, but what Gregor Mendel determined was purple flower color, that gene is dominant. That gene is dominant over white flowers. And really that all that's happening here is that the purple colored gene, that DNA, is being expressed, right? It's being read by the cell and it's saying, let's make purple colored pigment. The flowers that inherit the white, they don't have the gene that says let's make purple. They don't have any copy. It didn't come in the egg and it didn't come in the pollen. They don't have any copy of that information on how to make purple. So they just make white. So they make the lack of color, which is white, right? Blue eyes is really the lack of melanin, although people who have blue eyes make melanin in the background, but they don't make it in the foreground of the iris. So it's the lack of the color, right? So that's the basics of what Gregor Mendel did. And here, if you look in your handout, I'm just going to go forward to this page right here. I'm going to go back one and then I'll fill it out for you. But really what Gregor Mendel did was he crossed a purple flowered plant with a white flowered plant. And every time he did that, he got all purple flowered plants. So because he was a scientist, he was like, well, what's going to happen if I cross all of these purples that I just got? I got 100% purple flowers. What would happen if I crossed all of these? Like, is the white gone forever? Did it blend in there? That was what they believed at the time, that genes blended, but that doesn't actually make any sense because we would all be the same by now. You don't have to worry about that. But I think the reason why Gregor Mendel's information was not picked up for like 75 years is because people believed that genes blended. They didn't know that they were discrete packages of information. So he basically took two of these purple, what we call F1 generation. He took those purples and he crossed them. He did the same thing, cut off the filaments, did the whole pollination thing. He crossed them, and the next generation got a white one back. Wow. I'll show you how that happens. Oh, there's my puppy. I'll show you how that happens in a minute. But what happens is, and this is predictable. That's why genetics is so interesting, because there's predictable patterns. And he got 75% purple and 25% white flowers in the next generation. So if you have this page open, let's see if this will work here. And so P generation just means the parents, right? The parent generation. And in our situation, the parent generation was purple. Oops, let's see if this purple flower crossed with, I have to learn how to write better, huh? White flower. Okay, so that was the So that was the parent generation. And then once he did that cross, he then had the F1 generation, which is basically, it's called the filial is what the F stands for, but just think family, right? It's the first family that comes from the parent generation. And when he crossed the white and the purple flower, he got 100% purple flowers. I'm trying to turn that off, but I don't seem to. Let's see, hide names of annotators. 1 second. There we go. Okay, so he got 100% purple flowers. And then in the F2 generation, that means he crossed 2 of the F1 generation plants, right? He took 2 purple flowered plants and he crossed them. And then from that, in the F2 generation, what he got was 75% purple and 25% white. Okay, so that is the outcome of Gregor Mendel's experiments that really truly led to him understanding the idea of dominance and recessiveness. He did this thousands and thousands of times. You can go like on his website and see some of these the books and the journals of information that he collected. Let's see here. Okay, try. There we go. So here's just an example of all the traits that Gregor Mendel took data on. He looked at the seed form, seed color, the pods, the color of the flowers, the position of the flowers, how long they were they tall or short plants, and he collected data. So he did this for a long time. He had a lot of data that he collected. From that, we get what we call Mendelian genetics. We're going to see that we're going to also study non-Mendelian genetics also. So we have to remember that when we talk about, this is kind of important. When we talk about Mendelian genetics, what we're saying is that in Mendelian genetics, when the dominant, I'm going to write this down because I think it's this important. So I'm going to just try to write it. When the dominant, I'm going to use this word allele. I'm going to explain it in a little while. When the dominant allele is present, the dominant trait, that's an F, trait, will be expressed. Okay, so I know that might not make sense to you right now, but it will eventually. When an offspring inherits a dominant DNA, a little package of DNA, a version of the DNA that's dominant, right? You inherit 2 alleles for any gene from the egg and the sperm. Maybe you get the dominant one in the egg, and maybe you get the recessive one in the sperm. You can get the dominant one in the egg and the dominant one in the sperm. You can get 2 recessives. So the version of the gene that you get is called an allele. Sometimes you get a dominant version, sometimes you get a recessive version. But if the dominant one is present, if you inherited one of those dominant packages of DNA on that gene, then you're going to express that dominant trait. That's called Mendelian genetics. Sometimes what I just said, sometimes this whole thing I just wrote is not true. It is actually not true at all. That's called non-Mendelian genetics. We're going to cover both of those. Okay, I'm going to stop this video now, and then I'm going to make another video on the vocabulary. All right, so I'm going to hit stop share. Let's see how well I do this. I'm going to hit stop recording. All right, I'll be back. Bye.